All right, good afternoon, everyone. Welcome to the structure connectivity session. So we have 4 talks today, and in particular, we have Professor Maxime Descoteaux from University of Sherbrooke and Professor Lauren O'Daniels from Harvard Medical School. They are true experts in the field of structure connectivity and tractography. So our first speaker would be Professor Maxime Descoteaux. He's a full professor from Sherbrooke University, and he's the director of Sherbrooke Connectivity Imaging Laboratory. I know Max from many years ago, and my first DTI paper is collaborated with Maxime. So Max, and also Maxime just co-founded the International Society of Tractography, and he's the first president of the society. So Maxime, please get started.
Thanks, Zhengwu. Nice to see you all. It would have been better in person. I'm sorry. Beginning of term here for both my kids and myself, teaching a lot. So I'm online. Happy to take any questions afterwards or by email. So today I'll be talking mostly about tractography, challenges and opportunities. And I played the game. I was given these requirements or tasks. So keep an eye out for yellow, for the open challenges in the next 5 years. The magenta or purple for things that could be improved in terms of data sets for the statistical community, so in purple, and in cyan or light blue, statistical methods that need to be improved in the coming years. So I know this is recorded, so if you watch this again, keep an eye out for these colors. And because I'm the first speaker in this session, I wanted to just in one slide remind the structural connectome pipeline, usually starting from diffusion weighted imaging, MRI, lots of issues there, lots of open problems there. It's not going to be the focus today, but basically nowadays at every voxel, we can reconstruct local information about the white matter structures. And we are now quite robust to crossing fibers. We can very well capture how fibers cross. We can have different orientations for different fiber populations. And then the process of tractography is an algorithm that will integrate these directions together to create tracks or streamlines that connect gray matter together. So these are virtual reconstruction of pathways that are essentially following the traversal of following the water molecules traversing our diffusion MRI data. Nowadays, most tractography algorithms also use an anatomy, a T1 weighted image, which gives us contrast about white matter, gray matter, CSF. And so several algorithms now use the notions of cortical surfaces. We have gray matter surfaces, subcortical structures, so we can use this to improve tractography. We produce a whole brain tractography results or a tractogram with millions of tracks, millions of streamlines. Then on the side, we also have parcellation algorithms that determine different gray matter regions, different nuclei. We put the two together and we build our connectivity matrix or our structural connectome. And here I want to stress that at the moment, I would say more than 90% of pipelines and papers put in this matrix a number, which is track count, the number of tracks between every pair of regions in the cortical parcellation. And you could imagine this not being just track count. It could be just a vector of features. So just to make sure everybody understand this, if I zoom here between two regions, regions 17 and 23 of the atlas, I have a U fiber. And so the number stored in that matrix is, for instance, 100 tracks. That's the number there. And between regions 17 and 34, that's another connection, another bundle. It looks like a frontoparietal bundle. Here I might have 52 tracks, and I put the number 52. So in the future, of course, we would want to be better than this and include something about the microstructure of the track, the size of the track, the features of the track. Because a lot of people are using the connectivity matrix as a proxy to the strength of connections. So is tractography a solved problem? No, it's not. I put a number up there in the corner. I don't know if you guys are familiar with this number, but it's estimated that we have roughly 100,000 miles or 160,000 kilometers of axons in our human brain. And these axons are at the micron level, at the micron scale. And unfortunately, our images are at the millimeter resolution. So you can imagine the resolution gap that, of course, will lead to several of our computational challenges. And so if you have one paper to take home with you from my talk is this one. It's a Nature Communication kind of overview of a challenge that highlights, even though it's now 7 years old, most of the problems highlighted in this paper are still very much open today. And I'll walk you through a few of them. And so in the 3 different colors, as you can see in the video behind me, as you can imagine from the 100,000s of miles of axons, first thing, it's really easy for tractography to get lost. So it's easy to create impossible tracks. Some regions of the brain are harder to track than others. And so this has a dramatic impact on your connectivity matrix. And tractography validation is extremely difficult. We don't have yet proper data sets, data annotation, labeled data to really solve the tractography validation issue. So you can see here in yellow, challenges, blue, better methods, and magenta, more data needed. So what's happening? Why are we getting lost so much? It's essentially because of this resolution gap, this crossing fiber problem locally. Well, essentially what you see here in the middle is that several fiber configuration globally can lead to the same local signal in the diffusion MRI data. So our tractography algorithm is lost. We don't know if we need to keep going straight, turn, cross, fan. And so if we take all potential pathways, we create huge numbers of invalid connections. And if we are too restrictive and we just take the most probable one, well, we have an underrepresented connectome. So this is known as the false positive problem, the invalid connection challenge. And so several groups have looked at this issue, and unfortunately, it's a bad issue for connectomics. So I highly recommend this second paper here from Zalewski's group, where they show that for connectomics and graph theory, it's a lot more problematic to have false positive connections than false negative. And so if your algorithm is creating too many false tracks, then your graph and your properties from that graph are more affected than if you're actually missing some connections. So this is something to keep in mind, and definitely something to improve in the coming years. I don't have time today. Now there's a few generations of statistical approach, mostly filtering-based approach that can help this issue. But there are very little generative approaches that handle this false positive problem. There are some positive take-home messages in this paper. One positive message is that true positives, so the large fiber highways of the brain, are well reconstructed by tractography pipelines. So that's a good news. Most of the large, well-known fiber tracks, language, vision, memory tracks are well reconstructed by tractography. Also, our processing pipelines do work. They help. We've shown that it's important to not just take the tractography from the console at the MRI, but to run a sophisticated pipeline does help tractography. So this is something we did well as a community in the last 20 years. However, the next generation of algorithm clearly has to be able to deal with this specificity sensitivity issue where we want to reconstruct everything as much as possible while controlling for false positive. And this is hard. So I would say methodologically, this is probably the most important problem for connectomics, and to build that connectivity matrix where you are sure that you get all true positive and at the least amount of false positives. So we need better methods that handle crossings locally. We need better methods that include anatomy. We need better global or hybrid methods that handle false positives. And if we do so, this will really improve connectivity mapping. I mentioned this at the beginning. Nowadays, we have more advanced algorithm using surfaces. So I believe a lot in surfaces in the future. These are hard computational objects. We have to deal with meshes, but with these meshes, we have information about shape, normal, curvature. And it turns out that we can improve tractography with surfaces. But even with our best algorithms using surfaces, I want to point out another challenge is we still have more or less 30% of the cortex that is unreached by tracks. So everything you see here in this image in purple, mostly at the sulcus, in the deep, at the low curvature areas of the surface, we have lots of regions of our cortex that are not even reached by tractography. So you have to keep that in mind. That means all these triangles or pixels of the cortex are not even touched by a tract from tractography. And not because they don't exist, it's just because tractography cannot reach them. So that's a nice seeding and termination challenge for methods development. A lot of areas are hard to track. I just mentioned the U fibers, the cortex, but also in the deep structure around the thalamus, putamen, the different deep structures, the cerebellum, in and out. This is probably a resolution issue of tractography, but you have to keep that in mind. What does that mean for connectomics? It means that our connectomes are underrepresented by these white matter pathways that should be present, but that are either missing or underrepresented. And they are overrepresented by just a large highways that attract methodologically all our tracks in the algorithms. Something to keep in mind, that tractography is not as easy or as difficult depending on where you are in the brain. And then, of course, the pathology issue. As we start using our algorithms beyond the human connectome projects, and we go to the aging brain, the developing brain, the Alzheimer's disease brain, or as we go towards lesion with multiple sclerosis or traumatic brain injuries, or any kind of pathology that creates holes in our masks, I would say today this breaks most of our tractography pipelines. Most of our pipelines have been developed for healthy brain without holes. And so we need better methods, better pipelines that deal with patient data. And this is a huge challenge for our community. Last but not least, can machine learning help? There's this nice review paper that states the state of the art and the current open challenge. The answer is yes, but we're not there yet. It's really the beginning. I like this figure. Machine learning can do lots of nice tricks with neural nets, with transformers, with RNNs, but at the moment, because of improper ground truth, improper validated labeled data, I would argue that most machine learning-based algorithms are as limited as our classic tractography methods. And so huge opportunity here to improve on the classical techniques. And so on one hand, keep in mind that on the left here or on your right, the unsupervised problem is much harder than the supervised problem. It's much harder for the machine learning algorithm to produce a whole brain tractography connectivity as opposed to but creating a few known white matter tracks. On the supervised side, this is like a segmentation problem. And we're becoming good and better at this. And most machine learning approaches here that we may hear from Lauren are actually outperforming classical techniques here. So with recurrent neural nets, even CNNs, autoencoders, we can actually cluster and segment the white matter in the known white matter tracks much better than the classical techniques. The biggest challenge is on the unsupervised way. How do we reconstruct a whole brain while reconstructing all the true positives, but controlling for the false positives? And to me, this is a data problem. So this is work from my lab, but we've been trying to build a database for machine learning-based tractography in the recent year. It's so hard. So many hours of manual quality control, cleaning up the mess from tractography to try to build different white matter tracks that are validated, annotated, and provide the proper scoring methods to train the next generation of machine learning algorithms. So this is an example here from my lab, but this took, thousands of hours of CPU, GPU, huge amount of data. And I truly think this needs to be tackled as a community, not just as a single lab. And so to conclude, with this tremendous complexity of white matter axons in our brain, this is huge opportunity and challenges for the community. More information, proper statistical methods are needed that can handle more data. I think we need to include more than orientations, microstructure information, myelin information, functional information, maybe metabolic information, vascular information, to have smarter algorithms that do not get lost in our grid. We need more anatomy, more anatomists, more rules to influence our tractography, and proper validation. More, but better also data, labeled data, and this will take the whole community to do so. So this is my last slide. We just created the International Society for Tractography, tractography.io. The membership is free. Please go ahead and we need you. We need statisticians. We need anatomists. We need clinicians to really shape the future of tractography and structural connectivity mapping. Thanks for listening.
Thank you, Max. So due to the time limit, we might allow one simple question. Yes, please.
Can you hear me?
Yes.
Okay, thank you for a very fascinating talk. I'm not sure if the question is simple, but what do you think is the future of machine learning when it comes to sparse objects in the brain? Like, say, if you take the brain of someone with small vessel disease, they'll have a lot of white matter damage and probably a couple of cerebral microbleeds that are sparse objects that are very difficult to model with deep and machine learning models right now. So how do you see the future of comorbid brain conditions where we have multiple types of pathology in the brain if we want to model that with tractography?
Yeah, I see your question is difficult to answer. For me, this is the future is probably a long-term objective in what you're just describing. Maybe as a short-term objective, I believe machine learning-based or statistical-based approach will leverage normative modeling type of approaches. So I believe that today, we could have good anomaly detection algorithms from machine learning or statistical methods that leverage large databases and detect at least where are the anomalies in these pathologies or rare diseases. And that would already be very useful. So where is it? Maybe not what, we don't know what's going on, but we know it's anomalous. And even for personalized medicine, I think that'll be the first step, probably. And then we can actually have better algorithms that try to understand what the potential link with symptoms and syndromes and clinical outcomes are. But as a first step, I think would be just an anomaly detection.
Thank you, Max. So due to the time limit, let's switch to our next speaker. Our next speaker is Professor Lauren O'Daniel from Harvard Medical School. Professor O'Daniel is an associate professor of radiology, and she is from computer science, and she got her degree from MIT. So please go ahead.
Thank you for that introduction. I'll be speaking today about defining and quantifying bundles. I'll talk about some benefits, some challenges, and new approaches, and try to fit it all into 15 minutes. So thank you very much for inviting me to give this presentation. So the outline of my talk, first I'll talk a little bit about what is a bundle, and then I'll talk about what should and can we measure from bundles, and some benefits and challenges when quantifying popular things like bundle connectivity strength and bundle tissue microstructure, as well as a quick overview of some new approaches like shape and geometric deep learning. So first, what is a bundle? Over here on the left, you see an elaborate dissection of a human brain. So that's showing an actual view of the white matter inside of a human brain, showing some popular bundles for studying. Now, in the lower middle, you see diffusion MRI tractography, which I think we're all familiar with, especially after Max's talk. But this is the only method that we currently have that allows us to do non-invasive mapping of brain connections in vivo. And what you see on the right, in the upper right, you see some anatomically named bundles. Like in the red is the AF or the archaeofasciculus that you can also see in the dissection on the left. And so these bundles that are shown here in the upper right come from our publication several years ago about an anatomically curated fiber clustering white matter atlas. And so what this is showing with large bundles like this, we're able to consistently parcellate these across the lifespan using machine learning methods. And in the bottom right, you can see another more recent publication by my group using machine learning methods to parcellate the superficial white matter of the human brain in an automatic way. So we have lots of ways to define bundles. These are some ways from my group that I picked to illustrate. There are lots of groups with excellent ways to define bundles, but we don't actually know how to do this in a perfect way. We don't know how to do this at multiple scales. Defining bundles is actually still a research challenge. And I'm also a member of the International Society for Tractography that Max just told you about. I'm a member of the scientific committee, and I'm the unit lead of the interdisciplinary definitions unit. And so we're just getting started, but our goal is really to get people together, including computer scientists, neuroanatomists, and neurosurgeons, and talk about how we define these bundles, and talk about also how do we define bundles that have mostly been ignored so far in the literature. So for me, this is a very interesting research challenge, probably less interesting to a statistician. So I'll get onto what the statisticians should be more interested in, which is what should and what can we measure from these bundles? So how can we quantify these bundles? My group has a recent review 2 years ago in Neuroimage about quantifying bundles, which is also often called tractometry. And so the main challenge here, one of the main challenges anyway, is that choices at every step of the pipeline are going to affect the outcome of the analysis of your data set. And so on the left, you see a numbered list of some of the main steps. Before these steps even happen, there are diffusion MRI, important pre-processing steps that are valuable to improve the input data. But first, there's diffusion MRI tractography. There are a lot of methods to do that. Then there may be corrections such as filtering of streamlines or removal of false positives. Then there's a step for segmentation of bundles, multiple ways to do that. And then finally, the part that we're about to talk about is the quantification of bundles, which has several options, and I'll highlight some of the main ones here and their challenges and benefits in this talk. But for more detail, to really drill down more into options at these steps and their potential effects with lots of citations, I refer you to our review paper that you see here on this slide. So the first of the really popular options is the bundle connectivity strength. And I put strength in quotes because, as Max described, it's a very challenging thing to quantify. I just have one slide on it, which I think is very complementary to the previous talk because you got a lot more information in that talk by Max. So the connectivity strength measurement is often called connectomics or network neuroscience, and it's fundamentally based on the number of streamlines connecting pairs of gray matter regions. So you can see in the image here, this particular image shows a macaque cortical parcellation. And then on the right, it shows a tractography connectivity matrix on the bottom and a connectivity matrix from quantitative histology on the top. And so this is a powerful abstraction. And on the bottom, we have some citations. There's a citation for the paper where this image came from, which is a nice Neuroimage 2020 paper. And there's also a citation to Dave Tuck's PhD thesis in 2002 at MIT. And that, to my knowledge, has the first connectivity matrix, the earliest connectivity matrix that I've seen. So it's been around over 20 years, this connectivity matrix or connectomics paradigm. So this enables really data-driven studies, network neuroscience. It enables comparisons to invasive track tracing, as you see in the picture, or to functional connectivity from fMRI. There are a lot of research challenges. This is an extreme simplification of the data. It's affected by false positive streamlines. And really, fundamentally, this measurement of connection strength is unsolved because axons are and will always be much smaller than voxels. So while this is a very meaningful paradigm and it helps study the brain, it's really always going to be a challenge to measure connectivity strength. So another really popular measurement is tissue microstructure within the bundle. So what is tissue microstructure? Tissue microstructure is based on mathematical models that are fit to the diffusion MRI data in the bundle. On the left here, you see a picture of a bundle. This is the arcuate fasciculus in image A, and it's colored by fractional anisotropy, which is a very popular tissue microstructure measure that ranges from 0 to 1, where 1 indicates very oriented diffusion, very different from isotropic diffusion, and 0 indicates isotropic diffusion. And so as you can see, this varies within the bundle. And this particular fractional anisotropy is a streamline specific or fiber specific measure measured from a multi-tensor model that was fit during tractography. But there are lots of ways to get the fractional anisotropy measured here. But what do we actually do with this? Most often we take either the mean fractional anisotropy in the whole bundle that you see in picture B, or on the far right in picture C. Another popular method is to look at the fractional anisotropy along the course or trajectory of the bundle, for example, from anterior to posterior and inferior as you follow along the bundle. So what are the benefits and challenges of this? The benefit is really that we can study the brain tissue within these bundles. This enables hypothesis-driven research. So one example of hypothesis-driven research over on the right, it's another picture from one of my group's papers, but there are many, many, many papers studying fiber tracts or bundles using tissue microstructure. So this particular image is showing that the microstructure, the fractional anisotropy within many of these bundles, many association bundles, has a significant influence on individuals' language performance as measured by a picture vocabulary score or task. And so you can see multiple bundles, the inferior longitudinal fasciculus, the arcuate fasciculus, and others have their microstructure or their tissue has a significant influence on individual language performance. And so this method enables hypothesis-driven research. It enables removal of false positive streamlines that are not in the anatomical bundles that are expected, but there are also many research challenges. One challenge is just choosing a measure from the wealth, the abundance of measures that can be computed from diffusion MRI. Other challenges are just how to do it. Should we do fiber-specific microstructure modeling during or after tractography, or should we just sample it from an image? When studying the bundle mean, we lose a lot of information. All of these options and choices can vary in specificity and sensitivity to whatever disease or brain variability we're interested in. And we don't really know yet what are the best options for any individual use case or application. And really fundamentally, we have limited methods to deal with this high dimensional data. So in a data set and a study, or even in one individual subject, you can have millions of streamlines and more than millions of points along them, or multiple millions of points along them. So there's a lot of data. And so how to handle that is really still a challenge. So what's new in bundle quantification? So one somewhat new thing is bundle shape. There's really still a challenge here because the tract or bundle shape and computational representations of tracts or bundles are really less studied in the field. You can see some earlier approaches from 2006 and 2009, where they had some models of the streamlines or models of the overall bundle shape. And these were used to study bundles. And then you can see a modern application in 2020. There's a new paper about shape analysis of the human association pathways. And this paper demonstrates that The shapes of the human association pathways vary in the population, and it goes along with a nice software, DSI Studio, which actually quantifies all of these shapes for you. So I think that's opening up a bit of interest in the field to studying bundle shape, because there's now a way to do it and without having to implement it yourself. But still, there's a lot of room there. And I'll just show one new thing from my group to give a taste of some interesting possibilities in the future. One thing we're looking into now is studying bundles using geometric deep learning. And this is my postdoc, Yuqian Chen's work. And so we're looking at the picture at the left, you can see that's what a fiber bundle looks like from the perspective of treating it as a point cloud, which is an object that geometric deep learning can be designed to handle. And then what you see on the right is an example result from our paper that's cited at the bottom that just came out in medical image analysis. So here on the right, what you see is a visualization of the left arcuate fasciculus. And these blobs that are in green and purple are representing regions of the arcuate fasciculus that are particularly predictive of individual language performance. So this is giving us a new way to attempt to analyze the data without so much simplification at the measurement stage, attempting to take effectively the whole data set through the pipeline. So I think these new approaches are very interesting moving forward. How can we really retain more of the data and study it? And definitely a challenge. So hopefully I was within the time. I tried to have my talk not be too long. Lovely presenting this to all of you. So to summarize, quantitative analysis of tractography enables the studies of the brain's white matter bundles, both in health and disease. And really, these are all topics of ongoing research. Both defining and quantifying bundles are ongoing research avenues. They have interesting challenges. And I think really that looking forward, new statistical and machine learning approaches have the potential to improve the way we study the white matter bundles, and they may improve our understanding of the relationships to brain function and to disease. So thank you to my group. Thank you to the organizers. Thank you all for inviting me.
Thank you, Lauren. I think you're doing very well in terms of timing. We have time for maybe 1 or 2 questions. So actually, I have a question for you, Lauren. So those
Sure.
are bundles, and they're really beautiful. I guess you have a very well and easy to use tools for segmenting for us to use them.
You are absolutely welcome to use them. Everything is open source, and definitely we can reach out and have a conversation. But yeah, they're definitely documented and reasonably easy to use. We ran them recently on the entire ABCD data set, and we've released measurements.
Wow.
Measurements from all the bundles in the first release of the ABCD data set. So yeah,
Great.
welcome for everyone to use them, for sure.
Great, thank you. Okay,
You're welcome.
So let's move to our next speaker. Our next speaker is Dr. Tengfei Li from UNC Chapel Hill. Dr. Li is currently assistant professor in the Department of Radiology at UNC Chapel Hill. So he'll talk about a Connectome-based spatial statistics based on brain functional well structure Connectome atlas. Okay, please.
Okay. So first, many thanks to Zhengwu's introduction, and thanks for all the audience who participated in this workshop. And today I will roughly talk about the recently developed a pipeline, the Connectome-based spatial statistics pipeline by our group, based on a newly constructed atlas. So this first, I need to mention that this is a joint work with Dr. Hongtu Zhu and Dr. Zhu's student, Xifeng Wang, who has graduated. And also, Zhengwu has provided a lot of help to this project. And actually, let me first introduce myself. I have been working with Dr. Zhu for about over 9 years. Yeah, and I can still remember about 7 years ago, we first applied for the UK biobank data, and then we set up all our imaging processing pipelines. And the most things that I learned from Dr. Drew's lab actually is how to learn from your collaborators. And we can, for example, do from the beginning, work from the beginning on the project and to the end. And we not only do the upstream, and we also can do the downstream tasks. Yeah, even though it may be not deep enough, but we can have an overall understanding of the how the project overall looks like. Yeah, and I think it's helping a lot in pushing the project forward. Yeah. Okay, so let me go next. Okay, first I will talk about the background and the challenges of the current fiber tracking method. And the second, I will give a brief introduction about our newly developed atlas and the CBSS workflow. And then I will give some preliminary results of the downstream analysis and the future directions. Okay. I think in Max's talk, he has given a very detailed and wonderful introduction for the fiber tracking background knowledge. And here I roughly give one page about the diffusion parameters and the diffusion weighted image. The white matter usually is the advantage is it connects the gray matter regions, and the diffusion parameters of the diffusivity may reflect the signal transmission speed of the neurons, and the diffusion weighted image can make use of its different b values and b vectors to get the different image intensities. So by fitting a diffusion tensor model or the ST equation, and we can estimate the diffusion tensor D, which can be characterized by a diffusion ellipse. And by estimating its eigenvalues, and we can get the diffusion parameters as functions of the eigenvalues, like the DTI parameters, FA, AD, MD, RD, and M. And besides these DTI parameters, we also can calculate the DKI parameters and NODI parameters using other methods of formula. Yeah. And to in the next step, how to handle these diffusion parameter map. There are usually two important areas, two important methods. The first method is we can directly calculate the white matter. The most well-known method is the tract-based spatial statistics, which has been proposed about over 18 years. And you can see that its citation has been it has achieved the 7,000, and so we can say that it almost dominates this field. And in the second method is we can do the fiber tracking and derive the brain structure connectome. And there are different kinds of ways. And I think the previous two experts has given very detailed introduction on this. And you can roughly take a look at the difference of the two pipelines in the two figures here. Yeah. And here I give a detailed comparison of the TBSS and fiber tracking pipeline according to my understanding. Yeah. And actually in this field, Zhengwu is an expert here, and I'm still new here. If any place about my talk, there is some errors you can point it out. Yeah. So usually the TBSS pipeline is super fast, and it can be completed within about 10 to 20 minutes. And the fiber tracking algorithm usually is it runs relatively slow. It usually costs about 12 to 20 hours or even more. It depends on different fiber tracking algorithms. And whether they can spatially align across population. And the TBSS pipeline, they register to the common template, and they can do the voxel-based analysis, the VBA. And for fiber tracking, they can do the fiber-based analysis of fiber points or connectome-based analysis. And for the reproducibility, for the TBSS pipeline is very high. is above 0.85. And for a fiber tracking pipeline, some is high and some are low. It depends on different algorithms. And for the heritability, I searched the existing literature. It seems like fiber tracking algorithm is around 0.2, and the TBSS algorithm can achieve above 0.4. And for other social agents, and the TBSS pipeline may have very strong signal with with other organs like heart imaging traits, like brain aging, like disease such as Alzheimer's disease, the disease status. And usually fiber tracking algorithm can be relatively noisy, even it has more signals, but the signal strength can be relatively weak. Yeah, and I think the TBSS pipeline next, it cannot link the gray matter together, and then it cannot do the structural function coupling, and it cannot capture the white matter fine details because it projects to the white matter skeleton, and then it cannot capture the graph topological structure. And fiber tracking, that is the advantage of the fiber tracking. I think if there exists some other algorithm that can combine the advantages of the two algorithms, the two methods that can integrate both advantage, that will be better. And this idea comes from Dr. Zhu. Yeah, and we developed this connectome-based spatial statistics that is called CBSS, and we will make a comprehensive comparison with the TBSS. Yeah. And here I show some examples. In the panel A, that is the reproducibility and heritability of different imaging traits. And you can see that the TBSS-based brain diffusivity has very high reproducibility, and the highest heritability. The heritability is around 0.5. Yeah. And in the panel B, you can see that the TBSS-based imaging trace has closely is closely associated with heart imaging trace and heart disease. And in the panel C, you can see that the TBSS-based imaging trace is closely related with the APOE and some brain disorders. And in the panel D and E, I give some other literature that has already calculated the heritability of the fiber tracking traits. Yeah. For example, in the panel D, this literature uses the FA to construct the connectome, the structural connectivity, and the heritability is below 15%. And in the panel E, it uses fiber counts as the structural connectivity. And the mean heritability, I think, is around 18.5%, 18.5%. Yeah, both of them are less than 20%. Yeah. And the major challenge is to integrate both advantages. Okay, 5 minutes. Okay, I'll be quick on this. Yeah. So it's a big data. The data is huge. If we want to do the imaging genetics, we have to use the UK biobank data. There's 60,000 subjects, and ABCD, HCP, all of them are very huge data set. And the HCP is high resolution, but the other data set high resolution is relatively low. And sometimes we cannot do fine-grained pasteurization of the brain. And the third difficulty comes out from the fiber. The fiber is its complex shapes will result in heterogeneous locations across the population, and it will bring the difficulties to align the fibers together. Yeah, and we developed this CBSS workflow here. And in the first step, and that is to apply the max fiber tracking algorithm to individually extract the tractography. And in the second step, we do the surface alignment to roughly align the surface, brain surface across the population. And then in the next step, we combine all the fibers across the population. There are about more than 1 million fibers, and we do the fiber clustering based on dictionary learning. And the tracked DL algorithm is very fast here. And in the next step, within each cluster, we can extract the central fiber to construct a BFSC atlas. Yeah. From the fiber clustering to the raw BFSC atlas, the central fiber is determined by the fiber that has the closest distance to the other fibers from the same cluster. That is called the central fiber. Yeah. And we use the population atlas of the HCP and MMP. This is high resolution, including 360 populations of the brain. And we also incorporate about 13 subcortical regions. Yeah, that's the first step. That's the first stage. We construct the BFSC atlas. And in the second stage, we project the UK Biobank's aligned TBSS output, the diffusion map, to the BFSC atlas. We can get the projected FA fiber map. And then we do the elastic registration to align the FA fiber map together. And next step, we can calculate its reliability, reproducibility, and do quality control to eliminate those ICC less than 0.4. Yeah. And in the last step, we can get a refined BFSC. And here are some details about each step, like using the fiber tracking algorithm. Here we apply this track the flow algorithm from the Max. Yeah. And here we do the surface-based registration. Here we can register individual grains to the common MNI surface. Atlas, yeah. And here in the next step, we do the pre-classing quality control. Like we can merge some neighboring RIs by using the ICC criteria. The ICC is determined by the UK Biobank's 3,000 test retest data. And we can process the image using test retest data. Each subject observed twice, and we can calculate the ICC. And eventually, out of the 360 cortical ROIs, 14 of them were merged. And plus the 13 subcortical ROIs, eventually we have 359 ROIs. Yeah, and here is some pre-clustering step, how we merge the different ROIs together. This is in the right is a network-based ICC. And we do the quality control based on this. Yeah. And in the next step is the fiber clustering. That is from the tracked DL clustering algorithm. Yeah. Here is an example how to, yeah, okay, cluster this, yeah. Okay, and in the next step, we do the skeletonization. That's one of the key steps, and we get the similar to the TBSS. They use the 2 mm dilated skeleton silent, and they project all the diffusion parameters within the silent to the tract or the fiber. And we similarly use the 1 mm dilated fiber skeleton cylinder. Yeah. And in the next step, we do the elastic registration to register the extracted FA across subjects. That is what similar to what Anuji proposed. Yeah. The elastic registration. We add some of the regularization terms into this objective function so that the fibers will not blow away when during the registration phase. Yeah. And here is some final results. That is the reproducibility and the quality control. You can see that the mean ICC equals 0.74, and after quality control, it achieves 0.76. Yeah. And here is one downstream analysis. We get a heritability of 0.33. And here we show the downstream analysis of the association with APOE gene. you can see that the general signal appears at within visual network and between visual and subcortical network. Those regions are negatively affected by the APOE gene. Yeah. And here is another downstream analysis. We do the trajectory fitting by combining ABCDHCP and the UK biobank data to do the lifespan trajectory. Yeah. And here is we make a general comparison. It seems like current version of the CBSS can incorporate both advantages of the TBSS and fiber tracking. Yeah. And I think that's all.
Thank you, Tengfei. Yeah, due to time limit, so we have to reserve the questions to the end. I'll be the last speaker. So my first name pronounced like Zhengwu. I know it's really difficult for many people to pronounce it, but feel free to ask me if you still have difficulty to do it. So I hold positions at statistics and operations research. That's my tenure home at UNC. I also hold joint positions at biostatistics, neuroscience, and psychology. So I've seen lots of really good talks about brain network analysis, and also we have seen very good talks on Riemannian geometry on surfaces. So today I'm going to bridge these two fields, and we can see there's a different way of analyzing brain connectivity, which I call continuous and atlas-free analysis of structure connectivity. And for functional connectivity, it carries the same idea. So thanks to my collaborators and my students. Without them, I wouldn't be able to finish the works I present here. I was super lucky to be able to collaborate with them, and I'll display their figures next to each project when I talk about the details. So thanks to Maxine, Lauren, and Tengfei, I don't need to introduce the background, so that saves me some time. Let's skip to the tractography stage. With Maxine's surface enhancer tractography, we can get connectivities to the cortical surface. So when you extract the endpoints of those cortical streamlines, you got a pair of points connecting different points on the cortical surface. Okay, so traditionally we've seen lots of analysis. I have to say over 95 or 98% of current network data analysis papers simplify the tractography into a connectivity matrix by using some kind of atlas. You probably have 20, 30, 40 choices right now to choose from. So you can see that it's problematic. First, you lose information at a sub-ROI level. And second, it's ad hoc in choosing which pixelation to use. Third, there's a limitation in resolution. Usually you have 50 to 300 pixels or regions of interest. Third, if you choose large pixels, you actually have ultra high dimension data, especially you got discrete networks. If you have 300 nodes, you're gonna have 300 square numbers in your connectivity matrix. It's very high dimension to model. So when you go to the continuous stage, the dimension is lower actually. So we get a new data object. We have those point cloud. When you have a cortical surface, you have pair of points on the cortical surface. So the domain is omega, S1, U, S2. You have left and right cortical surface, and you can inflate the surface into sphere. You get really nice geometry, and then you can do simple statistics, many statistics on the sphere, but you also got some distortions by inflating the surface, right? Points next to each other probably would be far away after you inflate the surface into sphere. But I mean, what's continuous connectivity? From the point cloud, we got actually, the first paper named this continuous connectivity came from Paul Thompson's group from 2000 17. He treat this the connectivities, the endpoints, as a stochastic process, point process on omega cross omega. There's a latent function U, it's an intensity function controlling the observed point you see. This formula actually bridges discrete to continuous. The integration of two regions of this U equal to the number of expected points connecting the two regions. So this is really nice function. And the U function here is latent function. It's called intensity function in point process, and it's called continuous connectivity. When you have multiple subjects, you can sample this U from a stochastic process, capital U, and you can also sample an integer. Given U and Q, you can sample those endpoints we have seen here. So that's the statistical framework we use to characterize the continuous connectivity. So what are the problems behind it if we really want to apply it to understand the brain? So here are at least the 3 problems. The first one is the estimation of this U on omega cross omega. The omega itself is the cortical surface. It's very complex. When you have the omega cross omega, you're at a 8-dimensional manifold space. And U, it has really complex pattern because we know our brain has really complex connectivity pattern. So when you have a complex domain, you have a complex function to estimate. It's not easy. The normal way of estimating is kernel density estimation. You can see it's gonna fail in this case. The second one is everybody has different cortical surface. So how to align cortical surface based on the connectivity patterns, based on those fiber bundles in the brain. We know fiber bundles, we share very similar fiber bundles in the brain. So these fiber bundles acting as really good landmarks to align on different cortical surfaces. But with the network methods, you will not be able to align them because you have a combinatorial problem when you have nodes instead of cortical surface to align. The third problem is how to link U to behavior. When you have complex function, right, how to do dimension reduction, how to do statistics over there. So I'm going to introduce, the simple methods our group have been doing to give you an idea. So welcome to, join us to do the awesome data analysis in this field. The data you see here, those are point clouds, and you have pair of points. The problem is try to estimate the intensity or density function on omega cross omega. The first method is to use kernel density estimation, the simplest one. You only need to come up with some kernel, define omega on cross omega, and then you do this simple summation and you divide by the n. But the problem is that it's very slow. You have millions of fibers and you have 32,000 vertices to represent the cortical surface. So put up these two together, it's almost impossible to compute or estimate the kernel density estimation. You have to do some tricks and so on. And secondly, you face some geometry distortions. In order to be able to fit some kernels, take heat kernel, you need to have your surface to have nice geometry, like on a sphere. On the original cortical surface, how to define a kernel, that's ongoing research problem. So the second effort we're doing is to define kernels on the original surface, and then we try to do some efficient estimation of the connectivity. So it's the same idea about kernel density estimation, but we play some tricks, we make it faster, we define kernels, remaining kernels on the complex surface itself. So that represents some improvement in terms of prediction, predicting the behavior traits and so on. Those remaining kernels can give us better results for linking the continuous connectivities with behavior and clinical traits. The third model is, we have a complex function, we have a complex domain. So in deep learning literature, there's a very active field in recent 2 years where you try to represent functions using neural network. So your domain is Rn, and you have your domain is Im. You have a function defined in this domain. From the coordinates, you can then represent the complex functions. It's called implicit neural fields. So that's another direction which we're trying to do. Compare with kernel density estimation, we have a more flexible, accurate, and faster methods to represent. In particular, the neural network rely on the composition operation to eliminate many biases when you try to feed the data. When you have very rough image with discontinuity and so on, using implicit neural field, you can overcome those limitations you have when you try to fit linear expansion of basis. So that's another direction we're working on. The second problem is to do cortical surface alignment based on connectomes. So we have this awesome fiber bundles. We know those fiber bundles existed in the brain. So from Max's talk, we know that they're valid fiber bundles. So they can act as landmarks for us to align different brain cortical regions, but in the literature, there haven't been done yet. So the goal then is to find a warping function omega to move the vertices on the cortical surface so that you can align the connectivity pattern of different brains. So we're going to use the continuous connectivity to do that, because once you use the continuous connectivity, you actually have a functional data alignment problem. You don't have a network data alignment problem. And then the functional data analysis align problem, you have many tools to do it. So we choose one and then just do the implementation. Although the implementation is very difficult because you have to handle really complex domains. You have to work on the diffeomorphisms and so on. So we have a very fast algorithm now for align cortical surfaces. The paper is almost ready. This is a mean deformation of the amplitude. You can see it's very interesting. Along this motor strip and visual regions, we don't need to do a lot of alignment or moving of vertices to align different people, but in the frontal lobe and those regions, we have to do a lot of movement. There's a lot of more variabilities across people. And then we also care after alignment if you can do better downstream analysis, say predict of cognitive traits or do hypothesis testing between groups and so on. We all demonstrate that this method can improve those downstream analysis. The third one is to link brain continuous connectivity to behavior data. We know that U itself, the continuous connectivity, is a function defined on omega cross omega. If we impose a grid on omega, say 10,000 vertices, and this matrix, it will be 10,000 times 10,000. It's a very complex function, very high dimension. So we have to reduce the dimensionality. A regular PCA on vectorized the U will fail due to memory issues. What we can do is we develop a reduced rank space where you have this K output product of K acting as the basis of this space, and then you can project this connectivity matrix, continuous connectivity matrix to the reduced rank space to reduce the dimensionality. And this reduced rank space offers a desirable trade-off between flexibility and the computational complexity. In particular, you only need to estimate K, which is a function defined on omega, which is not a function defined on omega cross omega. So the computation is much easier. And then compare it with atlas-based connectivity, you can achieve much better statistic analysis, especially when you try to link to behavior traits. You have a higher correlation in terms of prediction, and you can do better hypothesis testing between two groups because the high resolution you gain and the smoothness you impose in this continuous connectivity. And also this allows you, if you do this structure and functional together, it allows you to get a continuous structure functional coupling, which, there's another trend in the neuroscience, try to understand how structure and functional work together, interact with each other to help us in decision-making, many disease progress and so on. So this continuous structure connectivity we've demonstrated has much better reproducibility, and it can predict the behavior much better in terms of correlation and low mean square error. So in the last a minute and a half, I want to reflect on Jen's questions, and Jen has asked us three critical questions to us. What are the most important research problems? For me, focusing on this structural connectivity, I think Concon is an elegant representation. Right now, I just show you two cortical surfaces, and you can add subcorticals inside. Basically, your omega will be, the union of those cortical surfaces. So the mathematical framework gets stays the same, but your domain gets a little bit more complicated. And we need easy to use tools for analyzing and to demonstrate it's better than ROI-based analysis. Okay. The second one, I think, is data integration, the integration across time and scales and different modalities. If we care about the science instead of one image modality, right, we should look at all the data we have together. What is the relationship between different modalities, different times, different scales? I will talk about it in a minute. I think to do that, we need collaboration. For example, I'm good at structure connectivity. Isa is good at genetics and so on. If we all work together and then work on this large data set, we should be able to produce really impactful work together. And the third one is benchmark data sets. So people talk about benchmark data sets. What I want to add is that we need flexible and high quality benchmark data set. When we say flexible, we should represent the data in a format that we can do variety of downstream analysis. For example, structural connectivity. I think this continuous the point cloud is a good way of representing connectivity because you can do ROI-based analysis by count how many fiber curves between different regions. You can also do this continuous connectivity. You can choose different atlases to use and so on. By saying that, I mean that when you try to release data, there are common pre-processing steps you can do. You do this common pre-processing steps and then release the data into a format that people can use. They do least effort. They will do least effort to be able to use this data set. And also how the statistics community improve processing and share the data sets. Myself, I do a lot of pre-processing the data. I trust my pre-processing because I know what's happening behind. So I share my data as well. I think it's about an attitude. So everyone should be willing to share, and everyone should appreciate the data that we share because it's a huge amount of time, manpower, resources. Hong2 started process UK Biobank 10 years ago. He didn't publish any paper for 6 years. So after that, he got many papers. You can see that 6 years, I think 50 students probably. So it's a lot of effort to do that. If you want to get the data, so you need to get ready to appreciate such effort. And finally, data set pipeline papers. Data is very important. We should appreciate the data. So we should allow clean, high quality data to be published in high impact journals so that we can get those data and then use them. And so finally, how we can, as a statistics community, to do more impactful work. First, I think we can expand our research problems. We see a lot of statistical modelings, but data science life cycle is very long. It starts with domain question and data collection. So data is important. We don't have a lot of people working data collection. Given very limited resources, how to optimize data collection so that we can maximize the statistical power in downstream data modeling. So as a statistician, I think we have advantages to do that. This is a simple example. With different accelerations, you get different effect size. You can maximize the effect size within the limit you have. And finally, I think collaboration is very important. Especially, I want to praise Hong2's effort for hosting workshops and other formats for bringing us together so that we can collaborate. And there are fundings out there to support these activities. Myself, I have to try to get some fundings. We have workshops and summer camps supported by NIH. You're welcome to join us. But we all, as a community, should try to make the effort to try to gather people together to form those collaborations and so on. And finally, I want to say, be ambitious. In computer science, people talk about AGI, self-driving cars. They actually make it working. So what's the big ideas in our society, right? What do we want to do? Should we work on data analysis or should we try to solve real problems? I'm still working on this.
It's not self-driving car.
So I'm still working on this. I don't know, what's the big ideas in statistics. I'm trying to convince myself, something that works me to work for 10 years and 15 years. I need to find a problem like that. All right, thank you so much. So we still have time for, other speakers if you have questions. Yes.
This is comment. The first person to come up with the continuous connectivity is Keith Worsley, 20 years ago. He's the first person.
Okay. Yeah.
He developed what we call the correlation random field, which has continued. Then the Paul Thompson.
Mm-hmm.
He's a big reader of the Keith Worsley's research, so he got the idea from him and gave it to the Moyer, who is still on Paul Thompson.
Okay, I see.
Story.
Yes.
They
Thank
check
you.
whether Paul signed the paper or not. Okay, asking.
Thank you, Mo. Any other questions for other speakers? Yes.
It's more of a general question. I wanted to ask because a lot of the stuff in structural connectivity is based on what I call kind of gray matter driven connectivity. But there is also, the white matter driven connectivity, based on the tracks. What do you, in your opinion and other speakers, what do you consider to be more important as a kind of research area where you really want to connect it with health outcomes?
So that's a good question. I think we should look at the brain from this different perspective. Myself, for the last past few years, I feel like I was too focused on one modality. So different modalities has different advantages. Like structural connectivity, it's very reproducible, has good reproducibility, but it's not so well in terms of predicting cognition and some other behaviors compared with functional connectivity. Although functional connectivity is very noisy, but it's pretty good in terms of predicting some behavior traits. So I think we should combine this together in some really good mathematic framework. Yeah.
I think the structure functional combining the structure together should be the right way. Otherwise, how do they really link it together? Because eventually, at the end of the day, all these imaging data, we want to be translational. You need to link all the imaging biomarkers to the behavior and also disease. And that's the most important part. And the reason people take images is not just take pictures. And these days, basically, they wanted to develop an intervention or drug to treat these imaging biomarkers. And that's the why this is still very important. Otherwise, if we just take pictures, there's no link with the clinicals. That's practically why it's useful. And also you link it to with the tumors or the damage, right? This damage also linked with the clinical outcomes.
So if no other questions, we're going to conclude the session. Let's thank all the speakers again.